Okay, so I'd like to switch gear now, and I'm going to talk about NGN-401, which is the Neurogene gene therapy product. So this is a self-regulating gene therapy for Rett syndrome. So here are my disclosures. I'm Chief Scientific Officer at Neurogene. So in terms of the science, as we've discussed, Rett syndrome is really driven by having too little MECP2 in cells. But the MECP2 gene is a dosage-sensitive gene, and we know from patients with duplication syndrome and also from animal studies that too much MECP2 is also a problem. And therefore, when thinking about a gene therapy or a treatment for Rett syndrome, one really requires a balanced treatment goal where you're delivering enough to be therapeutic, but not too much MECP2 to cause adverse effects. So this is really challenging in gene therapy because of the biodistribution. In other words, the way that the gene therapy distributes throughout the body. So when you deliver gene therapy, the AAV virus has the gene encapsulated within the virus, which generates what's known as a transcript or mRNA, which then makes the MECP2 protein. But different cells take up very different levels of the virus. So you get some cells that take up low levels of the gene therapy product and therefore produce relatively small amounts of MECP2, but you have other cells that can take up a high quantity of virus and therefore produce too much MECP2, which is a problem. And therefore, we have developed a technology to try and overcome this. And this is using what we call our EXACT technology. Now, this is what's known as a single gene circuit. That is, it's a gene therapy that has an inbuilt thermostat, if you like, so that when it delivers the MECP2, it expresses that MECP2 at the appropriate level that's going to be therapeutic without driving too much MECP2. And the way it works is it delivers the MECP2 transgene, which goes on to produce MECP2 protein within the cells. But if there's a lot of the product gets into a cell, then basically there's a safety valve such that the excess mRNA is broken down. So this means that in different cells that receive different amounts of the gene therapy product, there's a sort of thermostatic control, if you like, in each cell. So we've shown that this works in cells and can also constrain expression in experimental animals. And if we move on to the Rett syndrome disease model, so these are mice that are modeling Rett syndrome, so they're completely deficient of MECP2. And this is a very severe model. So these animals reach a human endpoint at around 9 weeks of age. But you see, and these are the untreated animals in black here. But you can see if you treat with the gene therapy product, you're increasing the survival of these animals in a dose-dependent fashion. So the light green is the low dose, and as you increase in the dose of the gene therapy product, you get a larger therapeutic effect. And this is just survival. If one looks at the neurological phenotypes in this model, and this is a clinical score for Rett-like phenotypes, with things like abnormal breathing, locomotion, and so on, or movement. Then you can see that the untreated animals in black here become more and more severe, but the treated animals in green, you can see it's really pushing down the neurological score. So what we've shown is that NGN-401 produces a fairly significant dose-dependent increase in survival and a significant amelioration or reduction in Rett-like neurological phenotypes or effects. So as well as effectiveness, one has to turn to safety as well. So in this study here, we tested the tolerability of the gene therapy product. And this is now in female mice that are what's known as heterozygous. So they express MECP2 at normal levels in some cells and not others. So it's basically mirroring the likely clinical population. And what you can see is when we give NGN-401, it's completely tolerated. So all of the animals survive. Not only do they survive, but they don't go on to produce a toxicity score. So basically at these doses which produce the profound therapeutic effect, we're not seeing any adverse effect here. So basically, this is well tolerated. If you compare it to a conventional unregulated gene therapy, so this is the exact same gene therapy product, but without our exact technology, you can see that these mice drop out of the study because they're developing a severe neurological score. So this is basically overshooting the level of MECP2. So basically the exact technology that's embedded in our gene therapy product confers both enhanced safety profile as well as being more efficacious. And we've also conducted further safety studies in other species to show that NGN-401 is well tolerated at doses well above the clinical dose that will be used in the clinical trial. So in terms of the scientific conclusions, Rett syndrome's gene dosage sensitive disorder, and therefore a gene therapy is going to require appropriate regulation in order to be safe and effective. I've shown that the exact technology that's embedded in NGN-401 enables autoregulation of MECP2 protein expression, and that this provides superior efficacy and safety relative to conventional gene therapy approaches. I've shown that NGN-401 is well tolerated in rodents and in non-human primates. And NGN-1 is also delivering the natural full-length MECP2 to really maximize the potential for therapeutic benefit. So, and the route of administration is known as ICV, and this delivery approach is really selected to maximize the distribution of virus to the parts of the brain that we believe are most important for impacting the phenotypes in Rett syndrome. So in part 2 here, we have a planned clinical trial. So in January this year, the FDA gave approval for a first-in-human clinical trial. And this is going to be an open label trial. It's a single dose study, and NGN-401 will be administered by intracerebroventricular infusion. So that's an injection into the fluid-filled space within the brain. And the study population will be pediatric females with typical Rett syndrome. So the objectives of the study are as shown here. So this is primarily a safety study. So the primary objective of the study is to evaluate safety and tolerability of the single dose. And the assessments that will be looked at include an assessment of adverse events, neurological exams, clinical laboratory tests, and monitoring of potential immune response to the treatment. And then as a secondary objective is to obtain a preliminary assessment of efficacy, in other words, effectiveness of NGN-401. And this will be monitored over a 60-month period. And this will be through both clinician and caregiver assessments. Okay, so with that, I'm very happy to take questions. Thank you.